Hello everyone and welcome to my presentation, which is part of the sixth and final meeting in the INTRI consortium. My name is LeonieFRANSEN and I'm doing my PhD at Public Health England under supervision of Martin Leonard. So here's an overview of my project. The initial goal of my project was to develop an iPSC derived airway epithelium. However, due to difficulties in establishing a fully differentiated epithelium on the air liquid interface, the focus has shifted to primary airway epithelium as well as iPSC and CD34 derived macrophages and dendritic cells. We did some toxicity testing on the primary airway epithelium, which mostly involved RNA sequencing and PCR methods. We also did toxicity testing on CD34 and iPSC derived macrophages and dendritic cells, which mostly involved Tempo-seq and PCR methods. So these data will be integrated in order to establish cellular specificity as well as mechanisms of action. I'm currently working with Nicoletta at the Liverpool John Moores University in order to establish an ARP on nanoparticles-induced exacerbation of allergen-induced type 2 inflammation. For this presentation, I will focus on the characterization of the macrophages and dendritic cells, as well as touch on some chemical toxicity. So apart from the airway epithelium, I will focus on macrophages and dendritic cells, as these are known to play an important role in airway toxicity, mainly in inflammation mechanisms, and also in airway diseases such as COPD. There are different subtypes of macrophages as well as dendritic cells, and there are also different activation stages, which highlight the need of proper characterization in order to be able to fully interpret toxicity results. So here you can see an overview of the differentiation of the CD34 and iPSC cells. The CD34 positive cells were first expanded in ultra-low adherent cell plates and then further differentiated to macrophages using IL-6, MCSF, TGF-beta, and GM-CSF, and to dendritic cells using FLT3, SCF, GM-CSF, and IL-4. iPSC were first differentiated to monocytes using a protocol developed by the group of Sally Cowley and adapted at KU Leuven, where I did my secondment with Freya. And these monocytes were further differentiated to macrophages and dendritic cells using the same media corresponded to the CD34 primary cells. We used RNA sequencing analysis to characterize the CD34 differentiation towards dendritic cells and macrophages. We can see that some macrophage markers, such as Marco and CD163, are uniquely expressed in the macrophages. Also, some dendritic cell markers, such as CD1E and CD1C, are uniquely expressed in dendritic cells. However, the gene profile of these cell types suggests there is a mixture of cells rather than pure populations of macrophages and dendritic cells. This is why we used single-cell sequencing to further characterize these models. So on this slide, we can see the AbSeq part of the single-cell sequencing. On the right, you can see antibody histograms. On the top, it's the dendritic cells. On the bottom, it's the macrophages. The iPSC are highlighted in color, and the CD34 cells are in grayscale. You can see that some of the antibodies, such as CD1A, are higher expressed in the CD34 positive cells, and also HLA-DR. However, some other markers such as CD14 and CD163 are more highly expressed in the iPSC-derived macrophages. On the left, you can see the PCA and TSNE plot of the iPSC and CD34 macrophages and dendritic cells with only AbSeq markers. You can see that the iPSC do overlap some of the CD34 cells in the PCA plot, and that the macrophages cluster somewhat more together with the macrophages and the dendritic cells with the dendritic cells. Based on this PCA plot, there was also a TSNE plot made, and you can see some clusters that have yet to be identified. On the same samples, we also did full RNA sequencing of single cell. And based on the PCA plot of this, we can see that iPSC dendritic cells and the CD34 dendritic cells clustered together, and the iPSC macrophages and the CD34 macrophages also somewhat clustered together. We can then make a T/S&E plot based on specific highly dispersed genes, and we can see that the iPSCs are their own clusters, and there is also subclusters of cells which have yet to be identified. We have done some work in identifying some of the markers, such as CD11b and CD11c dual positive cells, which are mainly expressed in the dendritic cells, and CD14 and CD163 positive cells, which are mainly in some subtypes of macrophages and the iPSC. So some work needs to be done still to fully identify subtypes and clusters, and also to compare this to in vivo data to fully know which cells are present in these models. We did some toxicity assays for the N3 chemicals. So these are 24-hour Resazurin exposures for amiodarone, busulfan, and paraquat. The dendritic cells show more toxicity for amiodarone and paraquat, and busulfan didn't have much effect on either cell type at the tested concentrations. This was somewhat verified with PCRs. The fold change in dendritic cells is higher than in macrophages for CXCL8. However, for paraquat, the macrophage response was higher. So here's some overview of the Tempo-seq chemical exposures in CD34 macrophages. Busulfan didn't have much effect as expected. There is some effect for amiodarone, but this is mostly insignificant. And there is a lot of significantly differentially expressed genes for the paraquat exposure, such as CXCL8, MDM2, DASP1. So that's why we did ingenuity pathway analysis for this data set. We can see that some pathways are significantly upregulated, such as the senescence pathway, induction of apoptosis by HIF1, and the death receptor signaling. This is mostly expected from paraquat exposure. So we still need to do some work to fully identify cellular specificity and integrate all this data. We also have some data coming up for single cell sequencing of paraquat exposure in basal and differentiated airway epithelial cells, which could add to this information and make a better mechanism of action. So based on this work, there are some planned publications. First of all, a review paper on small airway susceptibility to chemical and particle-induced toxicity. Second, a paper on primary airway characterization and also differentiation effects on chemical toxicity. A small paper on iPSC-derived macrophages and dendritic cell characterization. And a paper on CD34-derived macrophages and dendritic cell characterization and susceptibility to chemical and particle-induced toxicity. I would like to thank you all for listening, and also everyone involved in the N3 project for their guidance and support, and most of all my supervisor for all the time he has put in. If there is any questions, please feel free to email me. My email is printed on this slide. Thank you.